Welcome to the Photosynthesis Chamber and Sensors Workshop, or webinar rather. So here we go. I'm just going to jump right in. This is a little short webinar. So I've already connected my carbon dioxide and optical dissolved oxygen sensors. Those are the sensors we're working with today. I'm going to switch cameras, so you're going to see a view of my setup here. And you'll see it looks really messy on this view, but then there's a little inset view in the corner there. Where you might want to, you can see a little bit more detail of what's going on inside of this chamber here. So you can follow my hand to see what's going on there. Now, if you're watching live, you're able to move the slider, rather the gray divider between each screen to change the size of either screen. But if you're watching the recording, then you're kind of stuck with whatever proportions you have there. So here we go. I've connected my two sensors already. Here's my carbon dioxide sensor. This is the dissolved oxygen sensor. I've got this box here, which I'll go over later. It contains all the parts that go along with the photosynthesis chamber. If we zoom in on the photosynthesis chamber, when you look down on the inside, you can see that there's a tic-tac looking piece in there. That's a magnet because it's on a magnetic stir. Since we want to keep the water moving, keep the gases distributed as any processes that occur in here might be happening. This is where we're going to be adding plants to be studying aquatic photosynthesis. So I've got here some sprigs of Alodia that I'm putting into the chamber along with that magnet on the inside. And the outer chamber is filled with tap water as well as the inner chamber. And the outer chamber is there to help insulate from temperature change whatever's going on the inside. I've got a light back here and I'm going kind of faster with the setup because once we get going with data collection, I'll go over in depth a little bit more about what's going on with these pieces. Let me turn on our picture here. So I'm going to put the lid on the chamber. I've got I'm going to measure dissolved carbon dioxide as well as dissolved oxygen. So I've closed the chamber. It's not 100% sealed. It's not an airtight seal, but it is closed enough to observe changes inside the inner chamber. So now you kind of have an idea of what's going on under the lid there. There's a mirror that slides into the chamber side. This is outside of the water, so that I can, when I turn on the light here that's in the back, I can reflect as much light as possible into the system. This is a dissolved oxygen sensor, and I need to take the cap off just by twisting it. While I'm looking at it, if I go clockwise, it takes the cap off, and I check that the cap is moist so that I know it's been stored properly, and there is a O-ring that helps the dissolved oxygen sensor get to the proper depth when seated in the lid. I'm going to turn on white light because we're first going to study photosynthesis. So the white light will be on, and you can't really see that it's on because everything's closed. But I'm going to take the stopper off so you can see that it is on. And you can see also on the side that the light is on. I'm going to turn on the magnetic stir. To begin, the system has to sit for about a minute for it to come to equilibrium before we start data collection. Okay, so my stir is going. I want to make sure my stir is not going super fast, but fast enough to get some motion at the top of the water there. And now let's look at SparkView. I'm going to share my screen. You should be seeing SparkView. And I've, again, connected my carbon dioxide and optical dissolved oxygen sensors already. We're waiting for equilibrium. I want to use a graph display in SparkView. So I click the graph template. And if you think, oh, you're going really fast, slow down. Bear in mind, I will share with you the directions that go with this investigation so that you can see how to set this up in more detail, how to get this display on SparkView, how to connect sensors, and so on. So here we go. I would say we've been sitting for at least a minute here since I've been talking. So I'm going to hit start, and we will see, hopefully make predictions of what's going to happen with carbon dioxide and dissolved oxygen when they're in the aquatic system with an aquatic plant with the light on. What should happen to my CO2 and my dissolved oxygen? Let's hit start and see what kind of trends happen. I'm going to give it a minute to show a little bit of response here. And while that data is being collected, I'm going to switch to a different camera view. So now you don't have that little inset view. Now you have this big, messy looking setup. Well, setup itself is just that part that you saw me putting the plants into. Let's go over the individual pieces of the chamber so we can talk about how they work. All right, so here's what comes with the photosynthesis chamber. The chamber itself comes with everything you see on the outside. On the inside of the chamber, there's that plastic case you saw that contained water and the Elodea plants. Got a whole series of stoppers. There's that little magnet right there for the stirrer that goes on the inner chamber. You got this piece that kind of looks like a mystery piece with another O-ring. Well, the small O-ring right here is what's on the dissolved oxygen sensor there. So that O-ring that went on that sensor, it came from the chamber kit. Now, if you're wondering about why didn't that come with the sensor, well, here's another dissolved oxygen sensor to show you how it's organized. The oxygen sensor, the dissolved oxygen sensor, comes with a narrower, more skinny O-ring. So we've added this a little bit more robust ring to help it seat properly in the lid on the photosynthesis chamber. And I talked about turning the cap clockwise to make sure I don't accidentally unscrew the top of the sensor. When you store this sensor, you want to make sure that that sponge on the inside is nice and moist, not dripping wet, but moist with distilled water for storage. And then when you're done using the sensor, you'd rinse it as if it were a pH probe or conductivity probe and replace the cap and put it on nice and snug. So the chamber, though, the sensor does not come with the chamber. The chamber is separate from the dissolved oxygen sensor. So we are getting some data here. We can see a change in CO2, oxygen. We're waiting for that to respond. Meanwhile, we are seeing possibly a trend. We'll keep our eye on it. So I'm going to change my units here while data is running. I'm going to change from seconds to minutes. My time right now is in seconds. If I click the time measurement, I can change it to minutes right here, because in the instructions for this investigation, it says that each run should run 8 to 12 minutes. And we'll just see how long we can run given our short 20-minute time together. So back to the chamber. So goes together as you see here. Now, once you get the internal pieces set up, then there's that light that you may or may not have noticed I turned on to get the white light on. Here's what it looks like on the back. It slides when the lid is on the chamber. It just slides on. Let me get this out of the way. The light fits in just like into this slot. And if you worry about putting it in upside down, it'll be obvious that you did it upside down because it'll be sticking up a little. Just flip it over to the right way. And let me show you what it looks like when you turn it on. I have a second one plugged in here. So if you're doing white light, here's what that looks like. Super bright white light. It's all LEDs. But the nice thing about this is you can test different colors of light. So students can do an inquiry investigation. Well, what happens to CO2 and O2 if I, instead of use white light, what if I use blue only, or green only, or red only? So there's that option to do a little bit more inquiry with students with the photosynthesis chamber, aquatic photosynthesis chamber. So that the power for that light, also it comes with many different types of plugs for AC in different all over the world. The other thing that it comes with, that mirror that I showed you earlier, there's a little plastic covering, protective coating that you would pull off the mirror before you use it. So all that stuff comes with the chamber, including this part, which I still have to talk about. This is a super important piece that comes with the chamber. This piece makes it possible to use the carbon dioxide gas sensor, which normally you use in air. Here I have another sensor to show you. You cannot put this sensor as it is in water. You will ruin it. It is not meant for water. It's for air only. But the chamber comes with the adapter that you put on the end of that CO2 sensor. Get it up there nice and snug. And then another product is the dissolved carbon dioxide waterproof sleeve. It comes in a pack of 5. You get these 5 sleeves that let CO2 in but keep water out. You would have to trim it to size, which is in the instructions for the investigation. So we make it about that size. Notice it's been cut, and there's instructions on cutting it. Before you put the sleeve on the sensor, put the O-ring on first as a tip to make it a little bit easier. Of course, students love this part. It's a great source of hilariousness for them sometimes. So pull the O-ring up until you get to the top of that sleeve, and notice how snug the end of the probe is down there, and that is seated into the system. And now we can measure dissolved carbon dioxide. So we're measuring dissolved oxygen and dissolved carbon dioxide. Now let's take a look at our data, and I'm going to scale it. So my run's been going for about 6 minutes, and we've already got some definite trends here. So I'm going to hit stop. And while we're going to be talking about the data, I'm going to turn off the light so that we can test what happens to these gases in the dark. And this is where students would make predictions. Again, what happens to CO2 and O2 in the dark? Now let's look at the trends here. The top is carbon dioxide concentration in ppm. And it went down. And hopefully that's what you predicted, because if photosynthesis is occurring, carbon dioxide is being consumed in the process of photosynthesis. It's one of the reactants to be combined to produce sugars and oxygen in the presence of light. Then we've got dissolved oxygen down here. If photosynthesis is occurring, we know that oxygen is a product, so we would expect that it would increase, which it is doing here in the light. So now this lab itself is written as a qualitative study rather than quantitative, but you could do some quantitative analysis if you wanted to. So here are some recommended tools. I would pick the coordinate tool, and it might be confusing at first. How do I do the analysis on each of these runs since they're on, I've got 2 lines on 2 different graphs? You turn in the coordinate tool, notice it went to the green run, and that is because in my legend, I've got the red box around the green run, which also says Y2, and Y2 is over here, dissolved oxygen concentration. And I would move my coordinate box over to the starting time, click the delta button, select the ending point to get the change in CO2 or dissolved oxygen concentration for me. Milligrams per liter is the same as parts per million in this aquatic system. So that's one way you can analyze the data. Another way you can analyze the data, now let's say I want to do run 1. I would have to switch to run 1 or Y1 in the legend. Y1 is CO2 concentration. I might want to check the rate or the slope or how fast the CO2 change in the first 2 minutes of the system's change. So in that case, I would toggle to the select mode down here in the toolbar. I would select the data that I'm interested in up to 2 minutes. And then I might want to use the slope, the linear fit, to see what's the, it's not a perfect fit, but it's pretty okay. I've got some variable data here. You might even want to just select between minute 1 and 2, because maybe we've got some equilibrium going on here and we're waiting for it to settle down. To see the M or slope or the rate of change, we've got CO2 dropping by 13.3 parts per million per minute. Whereas we could divide on the oxygen run, we could do the same type of analysis at the same time frame, or we could just take the entire run's change of milligrams per liter concentration of DO2 over time. Or, like I said before, you just look at, oops, pardon me, I accidentally removed one of the graphs. Let me put another graph on there. Oh, well, I accidentally showed you another way to add the measurement to the same graph. Now, I took away one of the graph displays. I can put that oxygen back into this single graph by hitting that button that I hit right there with the plus sign. If I hit select measurements and I choose dissolved oxygen concentration, I want the milligrams per liter measurement because that one is the one that's comparable to parts per million. And now I've got that data on the same display. So I should have started my run a while back. I'm going to start now. Let's see what's happening with the dissolved oxygen concentration and carbon dioxide concentration now in the dark. And it might be even more confusing now because they're wondering like, oh, I don't have 2 graphs anymore. I do have these colors. I could see that I have Y1 and I have Y2. So again, we're going to use the colors to figure out which data goes with which. So the bright blue color goes with Y1. I can go over here and look and see what Y1 is. So the bright blue, that's CO2 concentration. It's barely changing because look at my range here. It's from 580 to 582. That's only 2 parts per million. So maybe it's not changing at all. Let's make that scale look a little bit more reasonable. I can manually scale my data if I like and get an idea of what's going on. So now that I have a little bit more reasonable scale, I can see, maybe that's not much of a change at all. Or maybe it is going up, or maybe it's going down. We got to give it time to see what happens. But we've got on Y2, the orange run, Y2, that's dissolved oxygen. We've got a trend going here for sure. We can see that it's decreasing. And the range here goes along with Y2 for dissolved oxygen concentration. So now we can see I've got a trend for oxygen, but you know what? I'm not so sure what's going on with carbon dioxide. And here's the big reason why this lab was written as a qualitative analysis. Well, carbon dioxide is a lot more soluble in water than oxygen. So a little bit of a change is not going to show up as readily as a change in oxygen. It eventually will. So you can see that the CO2 is doing what you would expect very slowly. It probably is going up. We just need to give it time to see that trend. So you could see that trend over time. Look, let me change again our units from seconds to minutes so we can get a better idea of how much time has passed. It's only been a couple minutes. Remember I said the run should be between 8 to 10 minutes. And that's, again, because the CO2 takes time. Gases take time to dissolve in the water and exchange. So this lab really focuses on matter being exchanged between Earth's spheres. We've got gases from the atmosphere, carbon dioxide and oxygen from the air dissolving into the water. We've got the plant intaking some of those gases and also exporting some of those gases depending on what's happening. So here, when we're in the dark condition, this is where students kind of see the aha moment with what happens with these gases exchanging in a plant in the darkness and what happens with energy in the darkness. A lot of students might think, well, CO2 shouldn't change and O2 shouldn't change. Oxygen and carbon dioxide should not change because plants don't do photosynthesis in the dark, so they shouldn't be consuming carbon dioxide. Well, that part's correct, but that doesn't mean CO2 isn't going to change and O2 isn't going to change. What's happening in the dark? There's that other process of energy exchange. That's that exchange called cellular respiration. Plants and all living things have to do cellular respiration all of the time. If they're alive, that's what they're doing. So even a plant that's photosynthesizing is doing cellular respiration. Why? Because it needs to make energy molecules so it can keep doing what it does all the time, which is grow, repair tissues, reproduce, basically make all the molecules needed for life, for living, and for reproducing. So we've got evidence here that carbon dioxide and oxygen are always being exchanged with the leaves. We've got an idea of what energy is required to push it through in the light. We need light energy to push the matter through the photosynthesis reaction when there's light available, and when there's no light available, we see only respiration. And what's pushing the energy through? What's pushing those molecules through the respiration reaction? Well, that's going to be chemical energy when those glucose molecules are consumed by the plant cells who are eventually going to be making ATP to drive other metabolic processes. So big aha moment here. Both processes occurring at the same time in the light, but in the darkness, we see only respiration where CO2 is going up and O2 is going down. Okay, I want to hit stop here, and I'm going to turn off my system. We got some pretty good data with just a few minutes of a run. The last thing I want to point out is on the lid. This is just one way you could set the system up. You could also put a temperature sensor and stopper or a pH sensor into this hole, or you can inject nutrients through the lid. There's all kinds of investigations you could do, and the place that you can go to find those investigations is at at pasco.com. Look for the Photosynthesis Chamber by name or by product number PS3251. And when you get to the product page, it describes all the stuff that I've talked about. It gives you the specifications. If you go to experiments, this is the lab. Where is it? There it is, aquatic photosynthesis and dissolved gases. This is the lab I just showed. You could also use algae beads. You could also do cellular respiration with yeast, or you can do enzyme action with hydrogen peroxide breaking down. You could also find the dissolved oxygen sensor, which is a separate product down there at the bottom. You can go back, and if you go a couple clicks more, you can find the carbon dioxide sensor and the dissolved CO2 waterproof sleeve. Another thing I want to show you is where I got SparkView software. That's also on Pasco.com, and it tells you how to get the free app. Okay, if you're live, I'm going to hang around and answer questions if there are any after we stop recording. Otherwise, thanks for joining us today, and have fun.